Hello, my name is Dr. Blake Bextine. I am an associate professor of biology at the University of Texas at Tyler. And we are involved in the zebra chip system, working with potato psyllids, which are the vector of the pathogen that causes the disease. Our main role has been phylogenetics of the potato psyllid, but that's actually led us to another area, an exciting area of insect regulation, insect control, which is using something called RNAi, and that's well known as gene silencing. So our idea is to actually take the normal cell process where a cell has DNA that's transcribed into RNA. That RNA message is then transported from the nucleus and taken out into the cytosol, or the rest of the cell, where it goes to a ribosome and is translated into a protein. This is normal situation inside a cell. So our goal in our project is to downregulate certain proteins. So what that really means is that we're taking that central dogma, that sort of general cell process of DNA to RNA to protein, and we're interrupting it. And so that's why it's called gene silencing, because we're literally turning those genes off so that the protein product is never made. So what we've chosen to start with is something called a heat shock protein. And the heat shock proteins are involved in construction of proteins downstream. So what we've come up with is a way to stop that messenger RNA, or that RNA, from going to the ribosome and producing the protein for a protein that actually helps produce other proteins. So we're stopping this big cascade of events inside of a cell, which ultimately stops the life of the cell. At this point, we have developed some constructs of RNAi, and we are putting those into cell culture that we have in the lab, and we're showing some cellular effects. Basically, the cells that we treat with the RNAi construct look bad and they die. The cells that are given the control treatment or the buffer treatment look fine and continue growth as they should. Over the next couple of months, we're actually going to try this in insect colonies and see if our RNAi constructs actually cause mortality in potato psyllids, and if so, this may be a very realistic treatment for management of the potato psyllids.